Hey, Reef Builders, and welcome back to another session of Reef Therapy. If you get a chance, please subscribe so you always know when we upload new episodes. We've got a very special guest on the show today, someone who takes the difficulty out of coral identification, Mr. Russell Kelley. Russell is a science communication consultant with focus in marine topics. He helps companies and institutions with branding and delivering communication strategies across all media. His work ranges from scientific papers and educational tools to international TV productions and keynote speeches. Russell received the Australian Coral Reef Society Medal for Science Advocacy and leads the Coral Identification Capacity Building Program, which offers training worldwide. I hope to take that, by the way. We'll get into that. But Russell, it's a pleasure to have you on. Oh, I forgot to mention you were the winner of the 2022 Reef Builders Choice Award for the Coral Finder 2022.
That's right.
Which I have right here.
Yeah, honored and privileged, and welcome to you and your listeners, viewers.
Yes, thank you so much for being on. I want to talk about a whole host of things, but let's talk just briefly about how you got into this. I always feel like there's some story that's attached to childhood that kind of gets you into marine biology or wherever you're at today. Is that the case for you?
Absolutely. I grew up in I was in a suburb of a town. Oh, sorry about the sound there. A suburb of a town called Brisbane, which is the capital of Queensland, which is a state in northern Australia. And this town was like a large country town, and has now since become a city. And now I am much older, but I grew up on the coast, and there were little islands offshore, and they had corals around them. There was like a fossil reef, and the living corals would grow on this fossil reef, and I'd just go and collect them. And I joined the local Marine Aquarium and Terrarium Society, and I became a legend in my own lunchbox because I could keep these corals happy, and I had a lot of ketodons in my tanks, and but the truth was that this is the 70s. The truth was that I cheated, because whenever things weren't looking tip top, I'd just get in my dinghy and go out and bring in a water change and just do 100% water change. And then I realized that the ketodons liked eating the corals. So I'd just bring in more coral, and they would just eat the coral, and I'd get some more every time I did a water change. And so in the 70s, we had none of the smarts and the dosing and all that sort of stuff that you have today. But I had a tank with 3 or 4 species of ketodon and half a dozen species of coral, and it looked like $1 million. And but the truth was that I was just sort of plundering the natural environment, we were using, in those days, it was sub-sand filters. There was that was it, and charcoal. Yeah, so very different times. And
Yeah,
then
sure.
I kind of fell in love with scuba diving, and I kind of became a diver more than an aquarist. But my passion for hanging out with aquarists remains.
Yeah, I feel like you're not a kid unless you plunder the natural environment a little bit, right?
Yeah.
Whatever's in your backyard, and your backyard just so happened to be the ocean. So, which I feel like I have major jealousy over. But maybe you also have jealousy over the fact that I had squirrels and rabbits in my backyard. So I don't know.
Yeah. Well, we are just fortunate that we lived in a time, in a place where you could be in the environment. And, I know that no parent today would let me do the things I did back then, like they'd be locked up as, kind of negligent parents. But I could just disappear for days on end, disappear for a whole weekend, and no one cared, just as long as I was back in time for school on Monday, and it was adventures in what at the time was fairly wild country, and you know, we just were privileged to have those freedoms, and so I hope you did too, by the sound of it.
Yeah. Yeah. We just got my oldest daughter 2 guinea pigs as of today. So we're going down that rabbit hole right now. My son has a betta and a leopard gecko, and she brought it to our attention that she had no animals to care for. So after we went through all the whole like do the research, now kids have the YouTube Kids and the internet that they can go do research on. So she knows everything about guinea pigs at this point. So we made them do the research.
Do they, they amenable to the punk haircut sort of style? The ones that like they have the tufty hair, and she can
Yeah.
actually put bows and ribbons in them as well. Yeah.
These ones, the ones that we picked up were rescues, and they're a couple short hairs, so they don't have the crazy hair. But I know exactly what you're talking about.
Yeah.
And some of the
Well, okay.
guinea pig breeders actually breed for it almost looks like straight long hair now. That kind of, it's not the tufted one, but it's super long, like long enough that you could probably braid all of it. It's crazy. But I digress.
Yeah, well, the fashion pigs are coming. I'm sure the moment she gets aware of them, there'll be requests.
For sure, for sure. You guys so it's our fall here in the U.S. So you guys are in spring right now. And am I correct in saying that it's coral spawning season there?
It's that time of year when a young coral's mind turns to what's going to happen about a month from now. So the Great Barrier Reef is very long, it's 2,000 kilometers long. If anyone want to do the translation to miles, I'd be grateful, but let's say 1,000 miles easily. But it in the north is maybe only 60 kilometers wide on the narrow continental shelf. But as you come down to the southern Great Barrier Reef, that becomes a 300-kilometer wide shelf. And consequence of that is the water is warmer close to the land. And so the corals, as they are making their eggs and sperm, which they just naturally do coming into summer, the ones who are inshore, around the inshore islands, it's like a cake in the oven. The oven, that cake bakes earlier than the offshore corals where the water temperature is a degree or two lower. And so everything in nature usually cues to the moon or day length. And so the inshore corals are going to cue to the next moon, the October moon. And the offshore corals will probably cue to the November moon. So day length is something that tells biology that, hey, the days are getting longer, and it's a rhythm thing that's built into the deep biology of all animals and plants. But then what you need is something to sort of say, is everyone ready? Queue up at the starting line, and that is the moon and the tide, gravity basically, and then depending, there are 300 species of coral just offshore from where I live, and not all of them. People tend to think that there's some sort of mass explosion that every coral spawns at exactly the same time, but what actually happens is groups of corals and species will spawn at a particular queue, and every individual of that species or that group will spawn at that time. But then there'll be other ones earlier or later, or the next night, or the night after that, or even at dawn in the mornings. So a lot of people have only recently clued onto the fact that quite a few corals actually spawn at 4 a.m. or 6 a.m. But everyone, because the initial Big Bang coral spawning, mass spawning event was sort of 5 nights after the full moon at 9:30. Everyone was kind of diving around that, looking for the patterns, but the patterns are actually spread out over the month, over the tide and moon cycles.
Interesting. Have you ever witnessed that in real life?
Yeah, yeah. Uh, I made a film about it uh, in the Pleistocene in 1993. Um, and then worked as a natural history film producer for about a decade after that. And so I shot it on analog video. I shot it on DV, early digital video. I reshot it on digital Betacam, and then I reshot it on HD, and then I reshot it with the Japanese on 4K when that came out. And now I'm retired. I've retired retired from coral spawning shoots just because it's it's it, it's just it's just never a perfect knowable thing. And if you put yourself out there as a consultant, sort of saying, I'll show you how to film the coral spawning, and I don't need that stress. Yeah.
Yeah.
But yeah. So a lot of sitting around on the bottom, waiting for things to happen. But at the same time, you that's the best way to discover other things. Most people, when they go diving, they'll get in the water and they'll swim for an hour and they'll go round and round and round, then get out. So the amount of time you spend in one place is usually seconds to a minute. But if you are actually forced to swim around the same patch reef or in the same little, grotto for day after day, night after night, 24 hours a day, with a team of people, suddenly you make discoveries. And it's like everything with biology. Most of the problem is that we just don't actually spend enough time looking. If you spend more time looking, you usually learn. But it's perhaps why the aquarium scene is such a rich source of interesting information now, because you guys are looking at corals 24/7, 365 under lights, with lights on, off. You can change the lighting, you can change the currents. It's your perfect little experimental microcosms. And this is something that the sort of science world, that wasn't even possible 20 years ago, not in a reliable kind of way. But now, you've got all this incredible information coming from aquarists, and it completely revolutionized how everybody looks at and handles corals. It certainly didn't come out of the science scene, really. It came from passionate hobbyists.
Yeah. You mentioned using your eyes, and I know that's kind of one of the big things when it comes to your guides that you've put together over the years, when it comes to coral identification and things like that. And I think we might have mentioned this before, but the name is escaping me. But the gentleman that created my octopus teacher had talked about looking at tiny little microcosms of the reef, or just a patch of sand, and seeing the different microfauna and things that hang out there, and how much you can learn if you just pay attention to one area for 10 to 20 minutes a day, or whatever the case may be. And I thought that was so fascinating, because you could take that and you could do that with anything. Go in your backyard, you could even people watch in New York City and Times Square and see the same people every day. It's just all about observation. And I think that there's such a beautiful thing to that, right? Just observing and noting and taking note and noticing any kind of changes that happen. But I wanted to kind of transition to your guides that you've written over the years. And obviously, I think this is the most recent one, right? The Coral Finder 2022.
Yeah.
And it really makes it super easy, for anybody to kind of go out there and check out the wild reef, and then and at least get a good idea of what they're looking at, right? Can you take us through, I guess, the beginner's guide to using this, because it's a tool, right? You have to drive this car.
Yeah, it's so it looks like a book, but you're right, it's a tool. And so it's not a thing that you read in a linear way. it's a tool that you interrogate. And when Corals of the World first came out, which is the 3-volume, 9-kilogram, put it on a little trolley to move it around epic by Charlie Veron, it was an astonishing achievement and the result of his life's work. But to be honest, I just couldn't use it because here's the coral that I want to identify. Hang on. Here's the coral that I want to identify. Which volume do I open? Information was locked up by being alphabetical. It was A to Z, effectively, across 3 volumes. But what I needed in order to be able to use Corals of the World is I needed some way of telling me which volume to open. So I out of frustration, I sat down and just looked at all the shapes and did a lot of copying and cutting up shapes and stuff and moving them around on a table, and then figuring out that, well, we could do vases and we could do sheets and we could do branches and we could do fingers, and there's some corals are attached, but other corals are free living. Maybe that's a group. Some are like columns. And so I reverse engineered Corals of the World in a way that would just let me point to what volume to open up. And so that was this is the 5th edition, but the early ones, the first 2 editions, were printed on plastic, and you used to be able to take them underwater. And then
that would be cool.
Yeah, it was cool, and it was a happier, simpler time. And then, of course, molecular biology came along and just smashed everything into pieces. And this is twice as many pages as the original Coral Finder. And I also started working in the Coral Triangle more, and so I had the opportunity to do more photography of more things, and that's how we arrived at the present sort of solution. But there's always a danger in this, which is who is it really for? If it's for the motivated person to learn the basics, is it really important for that person to know about a rare regional endemic species or genus from East Africa or from Japan? No, it's not, and so there's a chance that I might actually take some stuff out of the next edition just because it might make it a better learning tool, a better teaching tool, if you're not being distracted by really rare and weird stuff. So there's a funny tension in designing these tools. So what you're hearing now is the science communicator in me wrestling with the reality is, like, I want to be the coral guy who puts out the next book and it's going to be bigger and better and you'll be happier because it's got more in it. But whether it's going to be the best teaching tool it can be is a different question. And I'm really wrestling with that at the moment. So, there's almost enough new science to justify a new edition, new names and things that are changing. So I'm just waiting for enough new science to be published to justify breaking it into pieces and then trying to glue it all back together.
Yeah. I don't have the guide, so I haven't had a chance to really look through it, but is it, I guess if you were taking it underwater, it was designed for live corals, right? Not looking at the skeleton?
Yes.
Okay.
Yes. And it's a guide to identifying coral genera. And so all of the species in a genus are represented in the way it captures shape and form. And so some people might be a little disappointed that it's not like a list of species, but what it does is it teaches you hard corals being highly variable in shape and form with response to the environment, this teaches you how the corals are pushing that clay around, right? The genetic clay that they have is being shaped by the environment, and this lets you search by shapes, and then you go, oh, so I know what that genus is, and you can then go to the index at the back, and you can look up where else that genus occurs in the coral finder, and you can teach yourself, oh, I didn't know this thing can be branching, or it can be a vase, or it can be massive. All of these things are the same animal. It's just the clay being shaped by different species in different environments. And that's the real contribution of the tool, which is it turns the lights on for people at the level where they want to learn. And I have to resist the temptation to just keep troweling in more and more because it actually sometimes makes it a bit harder to use.
Right, because this sounds like it's designed for me, because I'm very much the, ooh, pretty color coral. And that's about it.
Yeah. Well, and the thing is, you'll be captured by the corals, which you are obviously, and you'll learn. How do you say when you passively, like it's just going to soak into you, and you probably will find if you had a coral finder, you'd find yourself just picking it up out of interest and flicking through it and joining those dots and connections, and it's not like you're going to sort of, your interest is going to stop at, I'm only interested in the pretty blue ones. Your interest is other pretty ones as well. But you might discover that, inadvertently discover that, oh, these things are related. And that in itself is just
sure. That, and I'm starting over now. Is the coral names that I knew before, they all got redefined, and they're not those corals anymore. So it was like, I was super into corals. Then I had that time where I actually had to go to work and not play with corals anymore. And then I pay attention again. I'm like, what? Nothing is called the same thing as it was before.
Yeah,
it's fembrophilia now.
Right. It's just everything. Like, it was acans, and now they're not acans anymore. I'm like, no, that is totally an acan, and I'm always going to call it an acan.
It's incredibly frustrating.
It is frustrating.
I'm on team science, but I also find it extraordinarily frustrating when some of the changes do seem to be a bit arbitrary. And so you guys would know Lobophilia and Symphilia. They're big meandering corals, fleshy with huge teeth. If you see the skeleton bear, they've got these big serrated teeth on them, very distinctive corals. And the Lobophilias were always the corals with separated walls, and so like you have meandering valleys, but there's a gap between them. And the Symphilias were always fused walls, and was about 10 species of one and about 10 species of the other. And when I was training people, I'd say, well, has it got separate walls? Okay, now to go towards species, we can think about 10 concepts of things with separate walls. And same thing with Symphilia. Has it got shared walls? Okay, we've got 10 different species we need to try and understand and work out. But what then happened is the taxonomist said, oh, there's not enough molecular distance between these things, and we're going to combine those two genera and use the older name. And the older name takes precedence by their rules, right? So Lobophilia is the oldest name. And so now what you have is 20 species in a genus that everyone used to know what it was, and now no one knows what it is because everyone's confused. And then when I try and teach this stuff, it's much harder to teach because you actually have to go, I know that it used to be like this, but now they're all together, and now I have to separate 20 concepts under the one name, one genus name. Where it used to be much easier, like it's 10, and 10 of those, 10 of these. So there's an arbitrariness in the way that science works sometimes where you'd sort of think, come on, guys, just We don't really need to. There must be other more important things to do than combine these names, which are just making life harder. They were well-established names. There was nothing wrong with the names. It was really quite an arbitrary act, in my view. But
well, I mean,
again,
I'm all about the science too, but I'm not getting any younger, Russell. I can't be relearning the same thing over and over again. It's not going to work.
But that's the thing. When I was a kid, I had a photographic memory, and I used to have. I subscribed to Tropical Fish Hobbyist, and I had some of the early TFH books, and there was a series of books called Pacific Marine Fishes, and the Herbert R. Axelrod. This is the sort of 70s. When I was, aquarium nerd, and I was a teenager, and I had what was effectively was a bit like a photographic memory. And to this day, when I'm diving, I look at turn around, there's a butterfly fish, and I look at it and I go, oh, that's, Chaetodon, Anisochetodon auriga on page 78 of Pacific Marine Fishes, Volume 1, right? But now it's called, and so I still have this kind of relictual memory that just sort of flushes through, and then I go, oh yeah, and then that changed, and then it got changed again. And with the corals, some of these things have been changed 3 or 4 times since I've been a coral nerd, and more information than anyone really wants to know. But you can't unlearn this stuff once you know. So it drives me nuts on a daily basis that I have to go, oh no, and then that changed, and that changed out, and I can't stop myself from doing it. Yeah.
So we're going to blame the taxonomists for us calling things Holy Grail torches and whatnot.
That's right.
Yeah, and they're a fairly powerless bunch, so I think we're safe. Yeah.
I don't know. They might reclassify us. So,
well, we'll just start an anti-taxonomy podcast with we have the means of production here.
There we go. Look,
propaganda them.
They made Pluto. They made Pluto not a planet anymore, and Pluto doesn't care, right? It still does what it does. So that's us. Change the names. We don't care.
The Coral Finder 2022 is Indo-Pacific hard corals. Now, I know that you've been working on something very special, and from what I can imagine is quite the task to do an octocoral book. Can you tell us about the new book that you've got coming out?
Yes, the Octocoral Finder. So firstly, most people reflexively would know what a coral is, and would probably go, well, yeah, but also, corals are generally things with a hard skeleton, and soft corals are different. They don't have a hard skeleton generally, and that's perhaps where most people's general appreciation might start and stop. Well, the octocorals are a group of coral in the polyp clan, the polyp animal clan, and the phylum Cnidaria, and these guys are the octocorals characterized by having 8 feathery tentacles or 8-fold symmetry. And believe it or not, the hard corals are also known as hexacorals, and they have a 6-fold symmetry. But generally you can't see it. You can only see it when the tiny little larval coral settles and it first starts to build its skeleton. And the very first thing it does is it lays down a pattern of 6 skeletal elements. And that's where you get the 6 from. But then it goes 6 to 12, 12 to 24, 48, 90, and it just goes on and on. Very soon you can't see that symmetry anymore. But in the octocorals, there's always 8 simple feathery tentacles. The tentacles have what are little feathery things called pinnules on them. And that group includes the soft corals, the sea fans or gorgonians, the sea whips, and the sea pens. And so you have these radically different looking organisms, like they're totally weirdly different. But when you look at them closely, they all have polyps and those polyps have 8 feathery tentacles, no matter what. So you'd sort of think, well, that would be interesting and easy, surely. Surely we can identify these things systematically and easily. But the reality is that they're very different from hard corals for another reason, which is your hard coral has all of the polyp animals, the cnidarians, are all characterized by one other universal characteristic, which is the presence of stinging cells. And so nematoblasts or nematocysts is another name. In the hard corals, they have these little dots all over their tentacles, and when they touch food items like little plankton, they explode and kill them by putting a little thread inside them, and a drop of venom goes down, poisons the thing, and then they pull it back in and eat it. But they can subdue plankton up to several millimeters long. With the octocorals, they have stinging cells, but they are completely useless for catching plankton. Like, they can't actually grab anything and hold it and kill it. They're more sticky. And what they're doing is they're feeding on a different size class of material. They're feeding on the microplankton, the picoplankton, and the everything from viruses, bacteria, and little microparticles and tiny plant cells. And basically, those stinging cells on the outside of their feathery tentacles are like flypaper. They're catching particles, and then they're passing them along, and they go down a groove and then down to the mouth. And these corals live in the same habitat, but they're exploiting 2 different food sources in the water column. So they're competing for space, but they're not competing for the same food. Now, why does that make them harder to hard to identify, and why has there never been an octocoral finder before? If we all know corals vary with environment, hard corals, and that they change their shapes in response to that environment. But if you find a weird hard coral today, and you go back a day later or a week later or a month later, it might be a little bit bigger, but it looks exactly like this. But if you go and look at soft corals, quite often something that was this big when you saw it on a dive, you come back 3 hours later and it's this big. So they have this capacity to inflate, deflate. And why are they inflating and deflating? Well, it costs them energy to be pumped up and to be these kind of big resisting the current bags of tissue. And they're, like a gorgonian. It was like a wall of mouths with, many thousands of tiny polyps on it with these sticky, feathery tentacles. And the water's, sieving through that gorgonian. That costs energy to do. And particularly with the soft corals, they'll pump themselves up to be big when the tide is running hard. But as soon as the tide slows down and there's less food going past, at some point they switch off and they just go. So you go back an hour later or 3 hours later or 6 hours later, and the same octocoral will look quite different. And so this design doesn't work this as easily. So when you use the Coral Finder, you will get sent to a gallery page, and there'll be usually, come on, pick one up. There's 1, 2, 3, 4, 5, 6 genera here for your eye and brain to scan, and you pattern search and you go, oh, I like that one. Whereas with the octocorals, that kind of thing won't work. I have to account for all of the
so do you have
soft tissue variation.
So do you have photographs of it in its different phases where you've got it fully erect? And
yeah, so that's been the job of the last sort of 5 years. Was just going, oh my God, what am I doing here? I'm the only person on Earth who thinks that this is important to do. So that's what's been happening. And so I was just looking a few days ago. I've compiled all of my octocorals from all of my trip libraries. So like I do a trip to this place, go to Indonesia in 2014 and again in 2016 and 2018, 19, 20. And I've got all these field trips, and then I log all of that material. And what happened about 2 years ago was the big molecular review finally came out for the octocorals. So they've had their big revision now. And hopefully, lots of names have changed, but more so what's actually changed is which families do genera belong in. So things got shuffled around and evolutionary relationships were reviewed. But what is now the case is there's going to be a certain amount of stability for the next decade while they do more work. And only now is it worth attempting to do a octocoral finder. So I've identified all my octocorals, and I have 21,000 to choose from, 21,000 images to craft this tool out of. And
wow.
Yeah, and like there's only going to be say 2,000 in the book, right? So it's
so where do you even
but
start?
They've got to be sweet, they've got to be the sweet ones that tell you the story, because again, it's not a species library. It's not an account of this species looks like this and that species looks like that. It's about being able to put a genus name on these things, and I have to capture and account for all of the variation of all the species in a genus, and then all of this title weirdness that goes on with them as well, right? So it's actually much harder to do than I thought.
So I think hobbyists might find it interesting, because I know I did when I first learned this, but sarcophyton, gorgonian species, and I'm just choosing three, and like a pipe organ, all have 8
feathery
tentacles on each one of those polyps. So I could go from those 3, which in my mind vary a lot in how they grow, and those are all considered to be octocorals, correct?
Yes. And most people, I think, very naturally see the detail of things down to family level or genus level. So, I'm just trying to think what the a trade name for a Dendronepthyr is, but it's the really spiky thing that looks like broccoli with little spines coming out of it everywhere. People will look at that, and it doesn't matter what species it is, they'll know it's from the Nepheidae, that family that produces those kind of broccoli corals with spikes sticking out of them. And it doesn't matter if it's got 100 species in there. People will recognize the clay, as I say. You recognize the grain of the wood. That a pine tree has a certain grain compared to a cedar or a eucalypt or something like that. You can pick up and you look at it. I don't know what the tree was, but I know it's a pine, a piece of pine, right? Likewise, you look at these sorts of corals. Most people have a natural a capacity to do their own taxonomy, like a xenia, this coral that does the pulsing thing, right? There's a whole family of them, and there's probably who knows how many species, but people would naturally be able to recognize them to family. It's just what we're trying to do is now create a tool that lets you naturally be able to resolve them by eye at the genus level. And part of what this job of this book is, and it's a bit like the wrestling with this thing of like maybe take some stuff out to make it a better teaching tool. Well, likewise, the science can only take you so far. And the scientists will say to you, you can't identify that gorgonian underwater. You have to cut a piece off, bring it back, dissolve it in bleach, and look at it under a microscope and look at the sclerites, the little spicules or spines in the tissue. And this book will tell you what you can know and what its modern name, its up-to-date name is, but it's also going to say, if it looks like these things, you can't actually ID it by eye in the field. You'll actually have to collect a bit because it's actually a bucket. There's a whole bunch of things that look like this, and you can't resolve it just by eyeballing it. And so known knowns, but also known unknowns. And it's a bit hard to concept to get across, but I'm actually sort of saying you can't go there.
Yeah.
I've got a little symbol, a sea dragon symbol, and it means here be dragons. You just don't put a name on it. We'll put it in a family. We'll call it Parameciidae, right? That's how far you can go, unless you want to be a nerd, collect a bit, do the science, blah, blah, blah. But all these other ones, yes, you can ID, and here are the really good photos to show you how to separate them, and here's the really good tips to show you how to separate them. So it's going to be a funny book. It's going to be about 75% of it will be, you can do it by eye, and 25% of it will be, it's a bucket, and we know it's a bucket, and this is the name you should use unless you're actually going to go and do the work.
I remember, God, I mean, this is really old school, but the early guides, it depended a lot on the skeleton, right? It wanted you to take a look and identify the septa and things that you can't normally see when the coral's alive. So it was, get the skeleton, that's your way to identify it. So this is leaps and bounds ahead.
Yeah.
Or well, I'm just that far behind. Probably that part.
There's always a challenge between science and the communication of science. And most scientists are not being paid to do science communication. They're being paid to be at the cutting edge of whatever their field is. And if they stop and turn around and sort of take a year off, or in my case, if I include the field work, it's 2 years to.
Yeah. Well, I'd argue that even if they were paid, they're not good
they don't get paid to do that, so they don't. Yeah. Sorry, say again.
I was going to say, even if they do get paid to do it, I would argue that many of them are not good communicators, right? You have people that are just good communicators, and then you have people that are good scientists.
Yes, and that's true in every endeavor. Sure. School teaching and so on and so forth. Some people are naturals, and some people literally have an astonishing capacity to inspire, and to bring people into the thing. And I do a little bit of public speaking in that sense, purely because I find I'm out of rage. I find myself frustrated that, say, diving isn't. Most people who go diving do not go diving in the way that people would go for a walk and identify wildflowers and know their local woodland or their local terrestrial environment. Diving is still 50 years behind the way we interact with the environment on land, and that's incredibly frustrating. And for me, I do these. I run a course called Coral Explorers, and it's purely about interacting with a group of divers and sort of saying, okay, you're diving the most complex ecosystem on Earth, and there's thousands of species here. But actually, we're just going to look at the ones that have got holes. And so years ago, I wrote a book called The Reef Finder, and it has a visual index at the front, and it lets you search by holes. And
I'm not touching that one.
And it lets you, so you can sort out sponges and sea squirts and worms and mollusks and so on. And it seems crazy but I do these things, and the next night, we'd be doing worms, and the next night we do a talk, and it'll be on octocorals, and then you find that the following day, people came back and said, I saw those things that you showed me, and they're just simple visual clues. And those visual clues are often universal, like the difference between a sponge and a sea squirt. It doesn't matter if you're on a coral reef or in the Arctic or 6,000 meters at the bottom of the Marianas Trench or on a rocky shore in in Northeast America. A sponge is a sponge and a squirt is a squirt, and the differences between them remain the same. So there's these universal things that we could be communicating to divers and to aquarists that would open up that crack of light that means biodiversity isn't just this kind of wall of stuff I don't understand, but there are patterns to be seen and natural dots to be joined, and there's no Latin involved. Like you just we use nothing but common names and nothing but things that you can see with your eye. And when you do that, people go, oh, I'm getting so much more out of my diving. I'm getting so much more out of my interaction with the environment than I've ever had before. And why is it? Well, it seems to be something to do with me having the passion to create the tools that open that door a little bit. But I think what it's also is there's a blind spot in the industry that they're still in the A to Z. A book is an A to Z thing rather than a tool that you use to solve problems. And that's what wakes me at night and makes me shake my fist at the sky.
Okay, so I have some slides that I want to go through. Do you recommend?
Oh, okay.
Do you recommend that we do the structure of hard coral first, or do you think we just go right into the trying to identify these corals that you sent?
We don't have time to do a kind of a workshop-y sort of thing, but what I might just point out. So you're going to throw that image up on screen in the podcast, is that right?
Yeah, and I can do that. I can do that right now for our
reference.
So
I'll just hold this up. There's a diagram here, and it has 10 terms on it, and they're really just pretty simple. Like one of those 10 terms in blue is mouth. So that's highly technical, but you can see that there's septa and costi, and here we're referring to the little vertical skeletal elements in the skeleton of the coral. And you can see in that diagram on the right, the tissue is on top. Now, the way to approach coral ID is to be able to work out what's called the wall structure. And you can see that we have wall in there on the bottom left. And in this coral, you can see that the wall seems to be a gap between the polyp centers. So there's a gap between one polyp and another, and that's what's called a coral with separated walls. Now, many corals don't have that gap. They're fused together, and so that's a polyp with shared walls. And that's, if you learn that much, you've got about 50% of the problem solved. So I thought we might just identify a few corals using wall structure. There's a way more to it, but just to show you that the Coral Finder works with very, very simple ideas. Now, before we start, the most important thing to do is to ask yourself, where is the mouth? So if you can recognize where the mouth is in the tissue, then you know that it is likely that the wall is going to surround the mouth. So just orienting yourself to where is the mouth is the absolute first step. So we might start with that if you guys like. Oh, and sorry, on the left-hand side here, you'll have the cover page. It's called the key page. And you can see that with the branching corals on the bottom right of the glossary diagram, you'll see it makes a distinction between axial corallites, which are lead corallites that lead a branch and are a bit bigger and usually a different color. And in the top left, when you're looking at the key page, you can see it asks the question, if you're looking at a branching coral, does it have an axial correlate or does it not? And that is about as technical and detailed as it gets. After that, it's all just wall structure again and being able to measure the size of the coral. But you can see that there are these shapes and lifestyles, meandering corals that are, meander like a river or have a pattern that seems to meander around on the surface of the colony. Massive or thick, that refers to not the size. It's not massive, is not bigness. It's massive is a measure of whether the length, width, and breadth are proportionally equal. So a massive coral is basically might be a dome, but it would fit inside a cube, if you know what I mean. Length, width, breadth, height equal. Submassive means one of those dimensions is shortened, and eventually you transition to just a thick coral, a thick crust. And at some point you transition to the thin plates, and then there are the solitary ones as we go down the key page. Large daytime polyps, with their tentacles out during the day. That's a group that you can search by. Columns and so on. So it's a pretty simple system, and once you learn how to recognize wall structure, you're off and running. You're off to the races.
Well, let's try the first one here. So this to me, but I think in the hobby right now, there's a little bit of back and forth on what these like maze brain corals are actually, and I think there's a lot of Goniastria, Paragoniastria. You hear just maze brain, Platygyra. And I don't know how many of these the actual Platygyra we see in the hobby, but walk us through how we would identify this one.
Yeah, so where is the mouth? And if you sort of go cruising along the bottom of the valley, every now and then you'll see a little sort of puckering circle, and that is going to be the mouth. And what is important to understand about meandering corals is that they can have many mouths. So corals are clonal organisms. So this is all the same tissue, the same genetic beastie. All you've got to do is sort of say, okay, the mouths, I can see them and they're running along the bottom of the valley. So therefore, you could imagine that at night there's probably tentacles lining these valleys because the tentacles are going to be associated with the mouth. And we know from the diagram that this is a wall structure that doesn't have a gap between the two polyps. They're fused. So this is a shared wall. And very helpfully, I've put the distance in there. So if you were to go to your coral finder and enter meandering and shared walls, and then you want to use the size, right? And so we'd be going probably in the 4 to 10 category there, and that would be where you'd start your pattern searching. And you can see I've included some text here, which basically is the descriptive text for Platygyra. Now, Platygyra doesn't have, super duper tight characterizations, but one of them is septa usually alternate and walls tend to appear ragged. And so you can see as you run along the top of the wall, the septa are of different sizes, and they're often sort of offset or a different size, and so they alternate in shape and size and position. And at a distance, that makes these things look a bit raggedy. And that is, a characteristic that separates them from something like Leptoria.
Yes, so the leptos are smoother, right? Like they're a little bit more.
Well, they can also alternate, but what happens with Leptoria is that they don't alternate and get offset. So even though Leptoria does have subtly alternating septa, they are in the same, they show the same overall width. And so it gives you that neat zipper-like characteristic of Leptoria. Whereas with Platygyras, they offset a bit and you get a little bit of a irregular, raggedy, sawtoothy sort of pattern. And it's subtle, but it's real, you know. And so, you know, once you've had it pointed out to you that Leptorias are zippers, and that it doesn't matter how they alternate, they always, you know, constrained, the wall is constrained to the same width. Whereas with Platygyras, there's just this subtle alternation of size, shape, height, position. Once you've sort of had that pointed out, you know, the mystery of Platygyra sort of goes away.
So then how is this different from, let's say, a Goniastria? Because it's very similar.
Yeah, so
looking.
You can see down the bottom in the text there, it says Platygyra lacks the paliform lobes found in Goniastria and Paragoniastria. Now, short of doing my workshop or watching some of my training movies, basically paliform lobes are like knuckles or teeth at the bottom of the wall. And so each one of those septa plunging down into the valley, and you know, you can see as you run along the valley there, you know, hundreds of septa plunging down. Well, imagine a little knee or a knuckle at the base of the wall. And that's what paliform lobes look like. And if you bleach the skeleton, you'd see that they are effectively like a sort of a specialized tooth that comes off each septum. And if each septum has one, then you get like a row of these knuckles. And so they often refer to it as a crown of paliform lobes, particularly if the coral is a circle. You get like a crown of these teeth at the bottom of the thing. And underwater, if you waft your hand over the coral and make the tissue, you know, retract, you can convince yourself whether something does have or does not have paliform lobes. And so Goniastria and Paragoniastria are corals with really well-developed paliform lobes, and Platygyra is not.
That's so cool.
Cool.
Let's do a couple more.
Yeah.
I think this is fascinating. I'm gonna skip that one. Let's do.
And I emphasize that, yeah, like there are 1,000 terms you could learn associated with coral taxonomy, but the Coral Finder basically gives you 10 to 20. And if you just apply yourself, and like I said, you know, one of those terms is mouth. You know, if you just apply yourself to being able to recognize where is the mouth, what kind of wall structure I have, and you also need to know about paliform lobes, and you need to know about budding. And if you can just wrangle those ideas in your head, then all of the coral genera can be resolved with a Coral Finder. And you know, it's just a question of applying yourself, and a good, you know, the only advantage anyone has is whether they've got a good eye for detail. It's nothing to do with big long Latin words or anything like that.
Yeah, I like that you've simplified it like that. Okay, so here we have an unidentified coral. Raj, what do you think?
So to me, this is looking like a favia.
Okay.
Well, yeah, bam. No, we don't. But let's run through the system then. Where is the mouth on these corals?
Well,
on this
right in the center of these. Oh, I'm trying to point. There's no pointer.
Yeah. So they're in the center of the ring, aren't they?
Right.
And I can see, you know, if I'm looking right in the central area there and to the left, I can see that quite a few of those polyps actually have more than one mouth. That's okay. Now, what kind of wall structure do we have?
They've got their own walls, not shared.
Yeah, that's right. So this is clearly separated walls. And then we need some scale information. So someone has diligently put that photo, that scale bar in that photo. And the way you measure corals is you go from the top of the wall to the top of the wall on the other side by the shortest dimension. And so you don't measure, some of these are quite oval, right? You don't measure along the valley, you measure across the valley. And so if you do that half a dozen times and take the average, that's how a taxonomist would crudely measure that coral and say it has, a diameter of whatever, right? So these are centimeter scaled, so 10 millimeters between the numbers. And if you measure across the short part of the valley there, you've got something that's about a centimeter, haven't you? They're on average, they're quite large. So 10 millimeters diameter, and that's the information that you would use to go into the coral finder. Now let's look at the growth form. It's basically a massive lump on the bottom. When I say massive, I mean a lump on the bottom, and the equal length, width, height, breadth, depth sort of thing. So this is going to go into the massive key group, separate walls, and we've got a scale estimate of around about 10 millimeters. So you could probably go to that second slide.
There you go.
So what does the Coral Finder offer you? On the key page, and your viewers will have to remember we sort of showed you the key page, which was the front cover with the shapes and lifestyles, and you can see this is a screenshot from the massive section, and you can see corallites with separate walls, 8 to 20 millimeters. And that says go to page 2728. And when you go there, which is that other screenshot, you'll be just eyeballing it, but you'll choose one based on what kind of looks and feels good. And then you read the text, which I've screenshot onto the right-hand side.
Son of a bitch, the text says it's formerly Favia.
There you go.
These damn name changes are getting me still.
So, but
so I'm not wrong, but I'm wrong.
Yeah. Well, but the thing is, you will learn, right? And in the back of you can see the red numbers, the ref numbers. In the back of the Coral Finder are the references. Reference 13, 22, and 28. 23 are the references that describe the science that changed these names. So it's empowering in that you are able to teach yourself. You'll be able to be your own guide. Now, if you just want to know what the name is, you stop here. But let's say you kind of like, well, I want to know whether I respect that name. You can go and read the original papers, and so that's what the Coral Finder does. It's this kind of reverse engineering tool that lets you go backwards and forwards and sideways, and we really need to be careful here. We haven't confirmed our ID, so the text in bold, corallites form circular to oval tubes with separate walls. Well, that's a tick, isn't it? There's a mixture of circles and ovals with separate walls. So we've got that. What's the next bit of bold text?
Dominantly intratentacular?
Yeah. So this is an example where if you read on in the Coral Finder, that you'll find that I lied to you and that there are more than 10 terms you need to know. And if you do my workshop, it's actually in the book. But if you do my workshop, we do a section on paliform lows. We do another session on budding. And budding is the process by which corals grow. And that can be by making a 2 from 1, cloning yourself into 2, can happen by splitting the coral inside of the wall. And if you look at the scale bar and you look at where it says 2 on the scale bar and you go up, I'm going to use about an inch, maybe 2 inches, you can see that coral is in the process of splitting. And so this is called intratentacular budding. So the bud, the cloning, is happening within the oral disc, within the circle of the polyp. Now, other corals, like Astrea, is an example of a genus, has extratentacular budding. And the new buds, the new clones, turn up in the gaps between the coral, between the polyps. And so what you'll find is like a little baby circle showing up, in the gaps between the corals, and that they tend to have a more circular. Most of the corals polyps are more circular. In the corals that have intratentacular budding, you tend to get a lot more of the ovals. Now,
so
can
the
we go back to
Favia does the same thing, right? Favias do the intratentacular.
That was what defined Favia until its name changed, but nothing has changed, right? The species are the same, the corals are the same, nothing has changed other than the name. And so
the
now
name
it's
is
called Dipsas Australia.
So there's no Favia anymore, or is that something different?
Oh, well, therein lies the story.
Caribbean Favia, right?
Yeah, that's right. So the name, so when the molecule people came and started, looking at all the corals, they had to. They say, "Oh, we've got some names that need to change here," and they are, these scientists are, they're kind of, well, they're
dicks.
new age molecule types, right? But they are
totally
constrained by the rules. They are constrained by the rules of taxonomy. And so one of those rules is, for example, if the same species was described more than once during the 20th century, the name that applies is the oldest name, the first name. So knowledge advances, and people, Charlie Veron, was the first person who kind of looked at everything all over the place, and he was the first person to sort of go, or not the first person, but the person who kind of did the biggest revisions of things in the old era. And he would say, oh, well, these things are clearly the same species, but it's been described and published more than once. Someone described it in Japan and someone described it in, Fiji. The name that takes precedence is the oldest name. Now, what happened is that the name Favia was applied to, is it Favia fragram in the Caribbean? And that genus and got carried over to the Indo-Pacific. And when, 2 centuries of science later, there are 20 odd species of Favias. But the molecule people turn up and they say, actually, there's molecular distance between the Favias, the Atlantic Favias and the Indo-Pacific Favias. And so we're going to give a new name to the Indo-Pacific Favias because they're a separate genetic clade. They belong, and they've been evolving in their own space for, tens of millions of years. And so the rules were that the oldest name stayed valid, but only in the Atlantic, and all of the Indo-Pacific Favias had to be given a new name, and that's how we ended up with Dipsas Australia.
I don't like this game.
Yeah.
You don't like it because you suck at it, or you're really good at it. One of the two.
But just try spelling it. Like that. It does not roll off the tongue or out of the pen. It's a shocker, that one, Australia.
So, you said you wanted to go back to a different slide.
Yeah, so if you just go back to the previous slide, which had without the screenshots on it. So I've just ask your viewers to scan around and look at how many mid-budding intratentacular polyps you can see. And, I would draw your attention to the top left corner. You can see right up the top left there are 2 that are clearly in the process of cleaving and becoming 2. And that's, and then you have to sort of say, well, any of these polyps here are either they're about to clone or they have just finished cloning, right? It's a never-ending process. And let's say down the bottom right-hand corner, you can see the dome. If we were to take a saw and cut that vertically, how do you think that's represented through time, right? At the very bottom was a little planktonic coral larva, a coral planula, probably knowing the size of that, probably 20 years earlier, had landed on the reef and started building 6 little skeletal spikes, because they're hexacorals. Remember, that's the characteristic that defines the hard corals, a symmetry of 6. And then the first thing it would do is it'd make a clone, and then those 2 would make a clone, and then those 2 make a clone, or those 4 make a clone. And what you would have, if you cut that slice vertically through that dome, what you would just see is, through time, these things bifurcating. So, one stem going up and turning into 2, and then those 2 turning into 4. And when you get to the outer surface, it looks like this. But they're all going back through time. They're all coming back and merging back into a single one, which merges back into another single one, which merges all the way back to the very first polyp. So these things are colonies of clones.
I feel like a cross section of that might be a good piece of art.
Oh yeah, cool. Yeah. And they're really good with X-rays. And I got a lot of cool material which I show in the workshops, which is for explaining all this stuff instead of my kind of ham-fisted verbal descriptions. It's nice to be showing that process.
Yeah, I don't want to go any further. You have a couple more examples here, but I think we've piqued enough interest to maybe get some more people interested in the class, or at least the Coral Finder book, and then the Octocoral book coming up. But I find this stuff super fascinating, and it's approachable, and I appreciate that, because I feel like and we've talked about this on the podcast before, but there's almost like a pretentious nature that comes with taxonomy and the hobby. Well, you didn't know that was micromucin now? You're still calling it an acan like Raj? You don't want to be like Raj.
You dumb
things like
bastard.
that.
That's
no
not a
favia.
Like
no,
that's a dip pastry.
But I do appreciate that's the science communicator in you, being able to translate hard science to average hobbyist, and I very much appreciate that.
Definitely.
Well, no, thank you. And this is why. And I must mention, my wife is my partner in crime. She pays the rent when I take like a year off or more to do a project like this. And you certainly don't make money out of selling books. I make money out of running workshops. So buy a book and then do the workshop. But my partner Rachel Pears is a true believer, and we love doing this stuff. I'll do it as long as I can. One of the things that's going to happen is there's going to be an online, by the end of next year, there'll be an online course that you can just take, which will just be like 10 training movies, and there'll be one for hard corals and one for octocorals as well. So that gets around the problem of me either having to fly to North America or you having to fly to Australia to do my workshop. We do them Zoom ones, and they're quite good fun actually. But I've sort of realized that at some point I'm just going to have to put it online and make it available to as many people who want to do it.
Do you have any Zoom classes coming up this year or next?
Well, yeah, I'd probably just say the end of the year, or maybe January, which is sort of a low season for us, high summer, when we take a bit of a break from the heat and tropical cyclones and things. Mm-hmm. So I I just am going to finish this book if it kills me. And if I survive to the end of the year, so my deadline is the end of the year. If I survive the book, then I'll be very keen to do a workshop straight away. It's just I can't say whether it's going to be in the second half of December or the first half of January. But if that's a time frame that works for you, you just go to byoguides.com, and you just use the email form to express your interest. Because that's what I'll just put you on a notification list, and when the workshop actually comes up, you'll just get an email a month ahead to let you know. And we've done a whole bunch of these. Jeremy did the first Zoom workshop, which I was terrified of because I just thought, oh, you can't teach coral ID over Zoom. That's just going to be awful. But it wasn't. It was really, really interesting, and I just kind of thought I'm going to lose people, but I didn't realize that people were registering for the course, and they were having coral ID parties, and they were putting it on their big screen, the big plasma sort of thing, and people are there, kind of yelling their names at the at the TV. And it was very entertaining and enjoyable in a way that I had no appreciation of. And so again, that's what I think is great about the whole ACRIS scene, is that you've got a bunch of people who are purely motivated by passion on the large part. So.
Yeah, that's cool.
Very cool.
Yeah, I'm excited. I hope that you get that book done so that I can take your class, because I'm super excited about it. I didn't get a chance to do it last year. I think it landed both times around Thanksgiving and then around Christmas, so we were with family and I wasn't able to slip away.
Yeah.
But hopefully this arrives at a better time and I can join you. And Raj actually said that he was going to fly me to Australia to do that,
just to take the course, just for the course. He's going to go there, take the course, and fly right back.
Well, I do run courses locally here. And I have, from what you could only describe as science surplus, I have probably the largest private collection of coral skeletons on Earth for teaching. And so if you were to be in Australia, it would be good to try and make a workshop happen, just because there is that opportunity, that tangible opportunity to combine the skeleton work with everything that I do, which is video and live coral photos in high resolution. And it's the marrying of those two things, marrying the skeletal information and how it translates through the tissue, that makes you a better taxonomist or a better coral ID user, is understanding the relationship between the skeleton and the living animal. So the Zoom workshops works really, really well and will get you IDing corals, but the kind of luxury edition would be to actually spend a couple of days with the collection and just noodling around, asking every question that ever occurred to you, because there's going to be examples of everything that you ever wanted to see laid out in tables. So.
You said the word luxury, Raj, so does that pique your interest at all?
It suggests to me business class.
Yes.
Okay.
I shoved myself in
all the way to Australia.
I shoved myself in, Coach, the first time I went to Australia, and I don't want to do that again. So, Russell, I appreciate it. This has been a great learning experience. I hope that everybody has gleaned some sort of information they can take with them on their journey. If not, grabbing the Coral Finder book that you offer, also the Octo Coral book that's on the way, and maybe you take a class or something in the future. But again, we'll have all the links in the description below if you want to follow Russell on his journey. And thank you so much for coming on. Thanks for joining us on Reef Therapy.
Well, my pleasure.
Thanks, Russell.
This is fun. And let's do it again with the Octo Coral Finder, and how to we'll do a show and tell and let you guys practice your ID.
Sounds great. Sounds great. Thank you so much, Russell. And we'll see you guys in the next one. Bye-bye.
Okay.
Bye, guys.
Cheers.